The United College Distinguished Visiting Scholar Scheme is made possible by the generous support of the college's endowment fund. Under this scheme, United College is able to invite world-renowned scholars to Hong Kong to give public lectures and meet staff and students of the college and the university. This year, United College has much privilege to have invited Professor David GEARY from the University of Missouri as our distinguished visiting scholar in 2014 to 15. Without further ado, I would like to turn the floor to the moderator of today's lecture, Professor Lei Chang from Department of Educational Psychology of the Chinese University of Hong Kong, to introduce Professor GEARY and the topic of this afternoon. Professor Chang, please.
Hello, everybody. This year's United College Distinguished Visiting Scholar, David GEARY, is a curator professor and Thomas Jefferson Professor of Psychology and Interdisciplinary Neuroscience of University of Missouri. His research spans two distinct areas: children's mathematical learning and development and evolutionary psychology. His pioneering work in mathematics learning uncovers the cognitive process by which children integrate inherent arithmetic abilities and naive numerical knowledge into learning and solving symbolic formal mathematics. His book, Children's Mathematical Development, published in 1994 and still widely cited, was the first in this area. And he has been continuing with this line of work, as evident in his upcoming edited book focusing on the child development of basic number processing. His work in evolutionary psychology covers wide-ranging issues, from the evolution of human cognitive abilities, the evolution of human cranial capacity, the evolution of mating systems, and the resulting sex differences in social and cognitive functioning, to his recent work on the evolution of vulnerable trait phenotypes. This wide-ranging coverage, however, is meticulously detailed and comprehensive. Evolutionary psychology bridges with many disciplines, most notably biology, genetics, anthropology, and paleoanthropology. Attending to the technical details and accuracy across disciplines requires the level of intellectual prowess and discipline that few can attain, and that very much defines GEARY's work. But more than details, GEARY's work is known for its innovation and insightfulness. His work on soft cognitive modularity delineates the interaction between genetic constraints and developmental plasticity, and helps to resolve the long-standing debate in cognitive psychology over how the mind works in terms of domain-general and domain-specific cognitive functioning. This work has become part of the foundation of the rapidly developing evolutionary cognitive psychology field. Similarly, GEARY's work on the construct and the distinction between primary and secondary abilities lay the foundation of the emerging field of evolutionary educational psychology. Primary abilities, such as basic numerical and verbal abilities, the understanding of causal sequence, the understanding and distinction between animate and inanimate objects, and understanding of others' mind. These are known as naive physics, naive biology, and naive psychology, form the basis for the advancement of modern sciences, including the learning and teaching of reading and math. And finally, his work on mating is innovative. Whereas most researchers focus on mate values, mating preferences, and mating-motivated behaviors, GEARY focuses on paternal investment and the evolution of the family system. He's pointed out that whereas chimpanzees are our closest relatives by all genetic account, in terms of mating pattern and behavior, we shared a common ancestor that was closer to gorillas than chimpanzees. Right? Gorilla mating system provides protoform of human polygyny and monogamy. So he's now embarking on something new again that has not even emerged in the evolutionary psychology or evolution literature, the evolution of trait vulnerability. Let's welcome Professor David Geary.
All right, thank you, Chang. I want to say, first of all, I appreciate being your distinguished scholar for this year. I have had a wonderful time at your great university. You're very, very nice hosts, and I'm very impressed with your university, and I'm sure you appreciate the quality of education you're receiving here. But now, this is my latest venture in evolution. Evolution of vulnerability or trait vulnerability. And I have a very, very simple proposal, an evolutionarily informed framework for identifying sex-specific traits that are especially vulnerable. Let me turn my speaker. There we go. Are especially vulnerable to disruption by infectious disease, malnutrition, environmental toxins, childhood abuse, Alzheimer's, a variety of certain things. So I'm going to propose a way in which we can identify ahead of time which traits, which skills, which physical skills, cognitive skills, behavioral skills are most likely to drop out first when there's problems. And this applies to humans and lots of other species. Let me move that out of the way there. I'm going to first give you the rationale for why I'm approaching this. I'm going to give you the evolutionary background and framing for understanding vulnerability. What do I need to get out here? Oh, there we go. I'll illustrate the utility of the approach with non-human species, then with humans, and then talk a little bit about the implications for human health and development. Okay, I think the battery's going out here. So the rationale. There are literally thousands of psychological, biological, medical studies of the bad effects that disease, exposure to toxins, childhood abuse, physical trauma, chemotherapy, so forth, on people's well-being in many different domains. Very few of these studies, and I know because I've read a lot of them now, have considered the idea that males and females may actually be differently affected by these different stressors. So maybe chemotherapy affects males in one way as side effects, and females in another way. Same thing for other types of stressors. As an example, psychologists and physicians have been studying the effects of parasite infestation, so intestinal worms, for example, on children's cognitive development, social development, cognitive abilities for more than 100 years. I mean, there's a very long history of studies on this. I think the battery's running out on this. Let's see. With the exception of some physical traits, nearly all of the studies put boys and girls together, didn't consider whether boys were affected differently than girls by infection with the same parasites. Or they used variables that I'll show you won't be very sensitive to these differences. 100 years of studies, the conclusion is mild to moderate parasitic infections don't affect children's intelligence or their cognitive abilities. I'll show you that that conclusion is probably wrong. We see a similar bias in the study of premature birth, toxin exposure, childhood abuse, post-traumatic stress disorder, chemotherapy, environmental exposure to environmental toxins, and so forth. They assume males and females are biologically the same, throw them together, and probably as a result come to the wrong conclusions about the risks of these types of stressors. So we're underestimating the effects of stressors, and we're losing opportunities for sex-specific assessments of risk and interventions for those. Okay, that's the rationale. The model is based on Darwin's sexual selection, and this is a way for understanding sex differences in vulnerability. The associated traits are associated with competition for reproductive resources. So these are northern elephant seals. They beat on one another for access to females. The physical competition results in bigger males than females. We have the peacock, of course. This is involved in mate choices. So males that have a lot of these kind of eye patches here and the coloration of them are healthier, and their offspring are healthier as well. So females use that information to choose their mates. So these traits have evolved to be social signals. They signal the ability to tolerate parasites, nutritional stress, social competition, and they can't be faked. I won't go into how that works, but essentially they're honest signals of underlying health, vigor, and so forth of the individual. Okay, these traits can differ from males and females. It's not just male traits, as I'll illustrate here. And that means that the sexes can differ in which traits are more susceptible. It might be one trait in males and maybe a different trait in females. So sexual selection just generally is competition for mates and mate choices. But I think we can broaden that to include competition for other types of resources that have reproductive outcomes, like female competition for food is very important, and that's called social selection. These traits are under what's called directional selection. So typically, the bigger the better. So more intense competition is going to push the trait to get more and more elaborate and bigger. Their expression is more sensitive to social and ecological conditions than are naturally selected traits. So even though they're evolved, they've actually evolved to be sensitive to the environment. Whereas other types of traits, like having 2 legs or something, is very biological, but it doesn't vary and is not as sensitive to conditions. So genes, of course, influence the development of these traits, but its full expression is also dependent on early conditions, prenatal conditions, early developmental conditions, and current ecological and social conditions. It's like everything has to go right. The genes have to be right, the early environment has to be right, and the current environment has to be right. Otherwise, their expression can be disrupted. In nature, parasites, worms or viruses or whatever, is a very common cause of disruption of these traits. Social stressors, competition for food, competition for mates can disrupt these. Poor nutrition is another common effect. And in the modern world, you have toxin exposure. I have a little asterisk here because some toxins will disrupt the expression of the trait, so it's not very well developed. Other toxins are called endocrine disrupting compounds, and they can actually exaggerate the expression of the trait. But that comes at a cost usually of higher mortality and other problems. So even though it sounds better, actually probably is not. And then inbreeding, these traits have more additive genetic variants than other traits. So inbreeding actually affects them more than naturally selected traits. And that's only important because it helps us identify what these traits are. So these traits depends on the species. I'm going to give you some examples here. It can be physical, they can be behavioral, they can involve brain and cognition, or any mix of these types of things, and are do not just involve females. So here we have a female soy sheep, and you see the horns there. Some females have horns, some females don't have horns, and during times of food shortage, like a tough winter, the females will fight one another. They're not fighting over mates like the males do, but they're fighting for access to food. Those females that get access to food are healthier, and their offspring are more likely to survive the winter than are the offspring of other females. And so these horns also signal social dominance among the females. As I said, for both sexes, different traits can signal vulnerability to different types of stressors. So maybe exposure to parasites will mess up trait A. Exposure to poor nutrition will mess up trait B. And some traits will be affected by developmental stressors, and other traits can be affected by current stressors. So there will be all sorts of traits and signals in these species that signal sensitivity to a variety of different things, a variety of different stressors at different points in the lifespan. So overall, when thinking about these things, the best evidence for trait vulnerability is when the stressor has greater effects on the sexually or socially selected trait compared to, and I'm going to illustrate this in a second, naturally selected trait in the same individual, the same trait in the opposite sex, or the same trait in related species. So for example, if we look at male soy sheep, they have bigger horns, and horn size will be normally distributed, and the mean males will be bigger than females, okay? So that would be the natural condition. And so if you're comparing males to females, or if you're comparing male body size to male horn size, you'll see those differences. Then you expose the population to stressors. The sexually selected trait, horn size in this case, will be more strongly affected by poor nutrition or parasite infestation than the other traits. Or in this case, male horn size will be more strongly affected than female horn size. Both will be affected, but the effect will be bigger for males. Okay? Does that make sense? Okay. So physical traits can be directly involved in competition, as we saw with the northern elephant seals, or indicators of competitive abilities or benefits conferred by mates. So if you're familiar with birds, you know a lot of birds are very colorful, and carotenoids are biological molecules that they get from their diet, and they produce yellow to red coloration. The color of these traits, here's an example, that's a black grouse. The color of the comb there is dependent on carotenoids, which means they're healthy enough to get a good diet. If this bird is food restricted, has an infection, or is exposed to social stressors, competition or whatever, that comb, that red comb, will shrink in size and will become less red. Basically what happens is these molecules, these carotenoid molecules, will be released from the comb and used in the physiological mechanisms involved in parasite resistance and modulation of the stress response. So they're, in a sense, a reserve. So, and these things, these changes can occur very, very rapidly. You can have the fading within 15, 30 minutes or so. So it's a very good indicator of current condition. You see that with plumage coloration as well. This is an American goldfinch. Very cool. There are a lot of those in my backyard at home. Structural feathers are a little bit different. They're not diet-based as much. This is a this is a grosbeak, and they produce blue, ultraviolet type colors. And the coloration is based on the structure of the feathers. And so if this animal is having stressors during the molt, the weeks or months prior to the mating system, then those colors will be faded. And so that gives you an indication of condition in the near past rather than current condition. Different trait, different indicators. So color quality influenced by testosterone, diet, toxin exposure, social competition. So these are all studies that have confirmed that these stressors affect these this type of signal. It's not just birds. You see it in fish, insects. Here you have an orange butterfly. So if you restrict diet during the caterpillar stage, for example, or heat shock during the pupa stage, so the distant past from this animal's perspective, that will be reflected later on in the types of colors it develops in adulthood. So different traits can signal sensitivity at different points in the animal's development. Sometimes size itself is condition dependent. So we have the alpine ibex, of course, with the big horns there. Inbreeding, postnatal nutrition affects that. So you have poor early nutrition, it's going to affect body size. So body size will get a little bit smaller in males, but the horn size will get much, much smaller. So the males will put more resources into body development than to horn development. So males with small horns had problems growing up, and you can tell that by that. Same principle works with lots of other species. This is a stalk-eyed fly, so they have these stalks here. Nutrition, other stressors, and they just get smaller there for males. So very good research on these species. And it's not just males. This is the diamond firetail. This is a female. And the structural spots, that means structural feathers, these white spots here are related to immunocompetence. So females that have fewer of these spots or smaller areas, their immune system is not as well developed as other females. So it's not just males, it is females as well. So there's all sorts of traits out there that give you an indication or a sign of ability to deal with stressors either currently, in the near past, or in the distant past. So here's some studies that I worked on, and this is a deer mouse. And what we did with these guys is we gave their moms bisphenol A. Well, let me tell you first. The deer mice, they're polygynous. When the males become mature, they expand their territory, and then within their territory, they search around for females to try to mate with them. So the males have much bigger territories than the females, which will result in the evolution of better spatial abilities, maze learning abilities, if you will, for males than for females. Meaning that spatial cognition is sexually selected or should be a vulnerable trait in males, but not in females. So to test that, we gave their moms bisphenol A, BPA. That's just a compound used in making plastics and resins and other types of things. A very common environmental contaminant. We gave it at levels that the FDA of the U.S. Government says is safe for people. And then we looked at spatial learning and memory in adulthood. And what we found was that normal males learn to use spatial cues. So they're in a maze. The maze is surrounded by a curtain, and there's shapes at different spots in the curtain that can be used to kind of navigate in the maze. Of course, when you first put them in there, they're just running around because they don't know what they're doing. Kind of got a random strategy. And then they figure out, and they go in circles, and they search for their exit hole. And if they're really good, they can use these cues to figure out, well, their hole is right in this quadrant, and then they can use that, a direct strategy. So normal males will do that. They go from random to serial to direct strategies. This is actually a birth control pill, estradiol, which the US FDA requires to be given in these types of studies because it's known that it has toxic effects on males, and so that's what it did. BPA had the same effect. If we look at females, they're basically using the serial strategy here and here. No effect of BPA on the females. The estradiol tends to masculinize females, and that's what we found here. So the BPA disrupted spatial abilities in the males. The spatial abilities were more fearful, engaged in less exploratory behavior, and they would have to be less fearful and engage more exploratory behavior to search out for mates in a real-world setting, but had no effect on their weight, audition, or vision. So if you just looked at them, if you're a person, you couldn't tell that they were affected. We then gave a mate choice study, and the girls didn't like them. So whether the girls were exposed to BPA or not, they preferred the unexposed males to the exposed males in a mate choice. They probably detected it by olfactory cue. So something about, there was some other kind of smell signal that was indicating that there was a problem with these males. We then did a follow-up experiment with a cousin of the deer mice, the California mouse. In the California mice, the males mate monogamously, so they share a territory with females, so they don't expand out, and so they don't have better spatial abilities than females, and spatial abilities would not be sexually selected or not be a vulnerable trait in this species, even though they're very closely genetically related to the deer mouse. And so we gave exactly the same thing, prenatal exposure to BPA, same dosage by weight and so forth, had no effect on spatial learning and memory in adults, in adult males or females, had no effect on fear or exploration, but did, critically, affect territorial marking. So males will mate guard their females, and to kind of demarcate their territory, they essentially just urinate all around the edges of their territory, and dominant males do it more often than subordinate males. And so what we did here is, over the course of a week, we put the BPA-exposed males and control males together in a cage. They were separated by mesh, so they wouldn't physically fight, and then just looked at how much they did territorial marking. No difference first day. By the end of the week, the unexposed males were dominant, and the exposed males were subordinate, much less trait marking. So this is a sexually selected trait for this species. BPA knocked that out, but didn't affect spatial abilities. But for its cousin species, where spatial abilities are a sexually selected trait, BPA knocked that out. So we have a sex by trait by species interaction. Very, very sensitive study to the hypothesis. So if you know a little bit about the natural history of the species, you can predict which trait and for which sex there's going to be problems. So the critical point is that across all of these species, one concept can be used to identify vulnerability. Traits that have been elaborated through social or sexual selection are especially vulnerable to disruption by exposure to environmental stressors. Okay? So anything that's been kind of elaborated above and beyond what you'd expect just from natural selection is a target for disruption. These traits can be behavioral, physical, or involve brain and cognition, as I've said before. It just depends on the species. You have to know a little bit about the species. They can vary by sex and species. The sensitivity can vary at different points of the animal's life history. And different traits may provide different types of information about resilience at different points in the lifespan. So all species are going to have a number of these different traits. Some may be sensitive to the early environment. Some may be sensitive to the current environment. Now, that's just the background. Now, the interesting thing is we can now apply this model to people, and which is what I'm working on right now. I'm just going to tell you a very, very little bit about sexual selection in humans just to set it up. So sex differences in physical size males are bigger than females, is very, very consistent with physical male-male competition in our ancestors. Males beat on one another to some extent, more so than females in our past. There's just very little way to argue around that. You'll have to take my word on this. Other aspects of male-male competition indicate a bias to compete, to form competitive coalitions or teams, whatever you want to call it. Males have advanced visual spatial abilities, just like the deer mice. They actually, in traditional societies, males' traveling space is 2 to 4 times larger than that of females. Differences related to use and construction of tools, projectile weapons, navigating an unfamiliar territory. So these are just a few examples of traits that should be vulnerable in males and less vulnerable in females. Women don't beat on each other as much as men do. I think maybe they should. Just why not, right? just to make things a little more interesting. But they don't. But they do beat on each other psychologically, at least as much as males do, maybe more so, especially during early adolescence. And that's called relational aggression. It's a form of female-female competition. It's more subtle than males beating on each other, although women are very sensitive to it. And that involves using gossip, lies, social manipulation, dismissing other girls and so forth as a way of excluding them from the social group, getting them away from your would-be boyfriend or whatever. That type of competition should result in the elaboration of language skills, sensitivity to subtle nuances in facial expression, especially among other women, which there is a big sex difference there, sensitivity to nonverbal behavior, theory of mind, and how much information you gather on others. So physical competition will make bigger males than females. Psychological competition will elaborate certain types of brain and cognitive skills in females relative to males. Sex-typed play and other activities during development involves refinement and adaptation of these competencies to local conditions. So boys and girls create different environments for themselves that kind of mimic some of the ways they compete, at least when the ways our ancestors competed. But my point here is that sex-type play should be sensitive to disruption as well. So we can look at those. Now I'm going to start with girls and women because, is there a clock? Right here? Okay. I'll just keep an eye on things. I'm going to start with girls and women because when people talk about vulnerability, they typically talk about male vulnerability. And even Darwin talked about males being vulnerable. And if we look at premature mortality, that's true. But I think we can do better than that. So I suggested that female-female competition will result in the elaboration of language abilities, for example, in women. So let's look at language development in preterm infants. Males have higher mortality preterm, but those that survive and have similar APGAR scores, health scores at birth, you get some interesting differences. So in this study, a large number of premature infants were followed from birth to 7.5 years of age and then given a very comprehensive assessment of natural language, or to 6.5 years of age, and natural language abilities. Premature girls, roughly born at 30 weeks gestation, so a little bit premature, not super premature, were deficit relative term girls in 18 to 22 indices of natural language competencies. They were deficit in spontaneous speech, auditory discrimination, word fluency, grammatical structure of utterances, and receptive vocabulary. All of these things would put them at a competitive disadvantage when competing with other girls. Premature boys, relative to other boys, there was only one difference. So it affected girls much more than boys. Same result, another big study at 5 years old, this time in Sweden. Term girls had better language scores than term boys. Common finding. We get the reverse of that. For preterm boys actually did better overall language skills than preterm girls. So language is more vulnerable to disruption in girls than boys. This was a very nice study. They looked at natural language development at multiple times over 5 years. And we see here, if anything is at the dotted line, the risk is kind of what you'd expect based on term. Boys, so we'd expect about 10% of boys to be delayed in cooing, and that's what we find here for the preterm boys. But 22% or so of preterm girls were delayed in the onset of cooing, delayed in the onset of sound imitation, naming their body parts, learning, following directions, learning their first name. There's only one area in which preterm boys were more deficit than preterm girls. So we have pretty good evidence that there is a trait in sex-specific vulnerability, and it's language here, and it's girls and not boys. Do women have social cognitive deficits with exposure to other stressors? That's what we would expect. As I said, most studies that have looked at exposure to, say, man-made toxins, chemotherapy, child abuse, and so forth, don't report sex differences or don't give good measures. There are some examples in the Alzheimer's literature. I'm going to go through this quickly so I can go through the data. Basically, Alzheimer's disease is associated with declines in social competencies, language-related skills, facial recognition and memory, processing of emotional cues, and so forth. And from the perspective I'm arguing here, we would expect these competencies to drop out earlier in women than in men. And in fact, there's a general tendency in the literature for that. The more women in the study, even if they didn't report sex differences, said, well, 60% women versus 40% women, the more women, the bigger the deficits. I did find a number of studies that did report sex differences, and most of these found a variety of language-related deficits in women, and typically the deficits were larger in women than men. So let me give you an example of that. So fairly large-scale study here. This is just a very simple word retrieval. So can you name objects correctly? Typically, women do a little bit better on that than men. In this case, the controls, there was no sex difference. We see men with vascular disease and with Alzheimer's disease are disadvantaged. There's a deficit there, right? But the deficit is much larger for women. And the language deficit is much larger than kind of the screening scale that you typically use to screen for Alzheimer's. Another study, somewhat more complicated, looking at word learning, so associating pointer with desk, kind of random types of things. You'll learn these associations. How quickly do you learn them? Women generally do better than men, which is what we always find. Men are deficit if they have Alzheimer's, but we have a crossover interaction again. The deficit is larger in women. Trying to remember these words after a short delay and a long delay. Again, normal women do better than normal men, but we get this larger deficit in women than in men. So the word learning, fluency sorts of things are dropping out more rapidly with Alzheimer's disease and vascular disease in women than men. It may be that women are more strongly affected by Alzheimer's than men. And that is generally the case. So what I'm talking about here would work only with the early onset of Alzheimer's. But just to test that, in this particular study, these guys looked at working memory deficits in men and women with Alzheimer's disease relative to healthy controls. And this is the size of the deficit on the y-axis here. And basically, both men and women have moderate, about a standard deviation deficit in general working memory skills, which is a significant deficit, but there's no sex differences there. But now let's look at word learning and short and long-term retrieval of words. Again, the scale of the deficits go from about 1 standard deviation to up to 7 or 8 or so. The males have significant deficits here, about 3 to 4 standard deviations, but the female deficits are devastating. They really just drop out. These skills just plummet in females with the early onset, despite no sex differences in these working memory types of things. So it's a very trait-specific, sex-specific deficit, just like I talked about with the deer mice and the California mice. So physical size should be sensitive to early problems more so in males than in females, particularly in adolescence. Not only height, but physical fitness, lean muscular mass, a lot of cardiovascular types of things, some blood parameters, and so forth. I've reviewed lots and lots of studies on this now. Most studies of poor nutrition, disease, and so forth during childhood, where there's not many differences in physical development, reveal boys might be a little bit more sensitive than girls, but not a lot more. But in adolescence, where you see really the sexual maturation, where a lot of these physical sex differences really emerge, you see huge deficits. So this is just an example. This is height. These are boys and girls with sickle cell anemia, so they have blood problems. They're also prone to infection and other sorts of things. The white circles are untreated. The black circles are treated. And you'll see in childhood, this is average height. This is below average height. This is above average height. Now the girls are about average in height. Childhood, the boys are about average in height. When they hit adolescence, it drops out. They really are seriously affected during that period of development. And you see this with malnutrition, parasite infestation, lots of other things, a very common pattern. Stunting, more than 2 standard deviations below average for physical size. Very consistent pattern across developing countries. For every female that's stunted in height, you have 2 males that are stunted in height. If you look at athletic ability, cardiovascular ability, physical fitness, and so forth, you get exactly the same effect, maybe even a larger effect for some physical skills. Okay, so again, not language, but it's important. Spatial cognition, I'm going to go quickly through this so we have time for questions. Basically, not a lot of studies have looked at that. Those that have tend to show that males are more seriously affected than females, but I'm going to give you a couple of examples here. So one of the studies that actually looked at pretty good spatial ability outcomes was done in Chicago, actually, looked at boys and girls' spatial development in high SES, medium SES, and kids living in poverty, boys and girls. And what we see is with the girls, for map reading and mental rotation, poverty doesn't have any effect on their spatial abilities, whatever it is. For boys, though, high and middle are the same, no effect, but the poverty affects the spatial ability development of boys and has no effect on girls. So it's sensitive in boys, insensitive in girls. Okay, no difference in IQ for this. Gua, is that the correct? Gua. Okay, study done in Taiwan. Prenatal, this was really a disaster. Company inadvertently contaminated cooking oil with PCBs, so you have a toxin, and a lot of kids were prenatally exposed to these PCBs, and then a lot of these kids have been followed developmentally for a long period of time. This is performance on a spatial reasoning measure. See a little bit of a gap for girls, but none of these differences are statistically significant at every age. It's a longitudinal study. Exposed boys had lower spatial reasoning abilities than unexposed boys. Again, no effect on girls. The same test associated with parasite infestation, malaria in this case, inbreeding depression, animal protein deficiencies, all affect spatial reasoning abilities more in boys than in girls, and oftentimes has no effect on girls. Sex-type play during childhood, you're all familiar with that. Very large sex differences, 2 to 3 standard deviations in size. Boys engage in rough and tumble play, object-oriented play, building things. Girls engage in more social, familial types of play. I found 3 studies that have looked at developmental exposure to toxins and sex differences in these types of play. Unfortunately, the studies haven't separated out the specific types of play. They've just given overall measures there. But in any case, in every case, prenatal exposure to these toxins, and it's a different mix of toxins in each of these studies, so you can get somewhat different effects on them. All 3 studies found that boys were affected. Here, for the first 2 studies, male typical play was reduced, and the one study that probably had some EDCs, different types of toxins, you get exaggerated male typical and female typical play. In any case, the early social behavior of boys is disrupted. No effects on girls in the first 2 studies. In the third study, which had a different mix of toxins, girls were affected as well. In either case, disrupting these normal social developmental processes is probably going to have long-term consequences for both sexes. Sex-typical behaviors include risk-taking, ease of fear conditioning, how quickly you get afraid of things, your ability to cope with stressors, in-group competition, and so forth. And all of that should be more developed in males than females. So we know a fair amount about the stress system and the fear system. The hippocampus, amygdala, and prefrontal cortex is all involved in kind of staying calm under stressful conditions or being very sensitive to threats or risks in a variety of situations. There's some very interesting studies now that show during pubertal development, there's testosterone-related increases in the size of the amygdala and the hippocampus in boys. So these areas get bigger. It wasn't thought previously that the brain got bigger during adolescence, but actually these regions do. And they decrease in girls, maybe associated with the onset of more anxiety disorders in girls. And anxiety is just the flip side of stress resistance or exploratory behavior or whatever. It's basically risk avoidance. Childhood stress can decrease the size of the hippocampus. Circulating testosterone levels actually mute the reactivity of these systems. So a larger system and circulating testosterone make adolescent boys and men less anxiety prone, more prone to take risks, calmer under stressors, and so forth. So we see all these systems emerging during adolescence and probably related to male-male competition. So they should be sensitive to stressors. And so if this system is actually related to male-male competition, then disruptions of it should increase risk for example, combat-related post-traumatic stress disorder, as it seemed like a fitting example to look at. So post-traumatic stress disorder, you're exposed to some extreme stressor that is a threat to your life or the life of somebody else, and then you have recurring nightmares, anxiety attacks, and other types of things, a very disruptive sort of thing. So generally, the more combat you go through, the higher the risk. That makes sense. But it also turns out that a smaller hippocampal volume increases risk of post-traumatic stress disorder in men. And it's not that the combat exposure or the stress disorder shrinks the hippocampus. It's because if we look at combat exposed, you look at identical twins, one exposed to combat, one not exposed to combat, and the size of the non-exposed twins' hippocampus predicts the severity of post-traumatic stress disorders in the exposed twin. So they're smaller. So if you don't have these sex-specific increases in the hippocampus during adolescence, you have male-specific, you have increased risk of difficulties with post-traumatic stress. Another risk factor is related to personal and family history of anxiety or affective disorders. Now, we know that girls and women have more affective disorders, more anxiety and depression than boys and men. I've made this counterintuitive prediction that exposure to stressors will make both boys and girls more prone to anxiety disorders than their unexposed peers, but that the effect will be relatively larger in boys and men than girls and women. It actually seems to be the case. So these risks are exacerbated by exposure to toxins, prenatal malnutrition, and other childhood stressors, and relative to unexposed same-sex peers, the effects are bigger in boys than they are in girls. Almost done. So prospective studies. This is looking at a bunch of college students who were assessed. So I mentioned that men tend to form groups, cohesive teams, and that's part of the male-male competition. They compete in groups. Men who don't form the male typical social, the male typical social relationships are more prone to PTSD than other men. So if they don't form, if they don't bond with their other unit members, they got that antisocial personality, dislikes authority, that probably is involved in the bonding, predicted risk. But what's interesting is masculinity-femininity scores we'll see there, controlling for combat exposure and asocial personality. Males that were one standard deviation atypical in their interests and social behaviors had a nearly threefold increase in the odds of experiencing post-traumatic stress disorder as adults after combat in Vietnam. And I showed you previously that these sex atypical interests and behaviors can be related to prenatal exposure to toxins or other types of stressors. So we would expect that this may have a downstream effect on the ability, particularly for males, to deal with very, very extreme stressors. So, implications. Despite thousands of studies in literally decades, more than a century in some areas, of research on human vulnerabilities to a wide array of stressors, sex-specific vulnerabilities are not only poorly understood, is typically assumed by psychological researchers, medical researchers, and so forth, that they don't exist. Males and females are essentially the same. And so we have lots of studies that are probably not very useful. Sexual selection, and more broadly construed to include competition for resources other than mates, provides an a priori framework to identify sex and species-specific trait vulnerabilities. So vulnerabilities in particular types of traits. So we'd expect, for example, women's facial processing to be disrupted, just like their language processing with chemotherapy or Alzheimer's, but not males processing as much. So I've given a few examples here of how understanding sexual selection in humans may enable a more nuanced and sensitive assessment of sex-specific vulnerabilities to infectious disease, poor nutrition, social stressors, toxin exposure, and so forth. The approach, I think, should enable the detection of problems earlier than is currently available. So if language and social cognitive processes drop out more quickly in women with Alzheimer's disease than men with Alzheimer's disease, then assessing these skills will allow you to detect the problem much earlier than it's now detected, meaning treatments can be instituted earlier. If chemotherapy affects these skills more in women than in men, then assessing the toxicity of the chemotherapy can be done in a more nuanced and sensitive way than is currently the case. This will help us develop sex-specific assessment strategies and perhaps remedial strategies for a variety of different things, trait and sex-specific sorts of things. Stressors that affect naturally selected traits as much as sexually selected traits are probably especially toxic. So I've looked at some chemotherapy effects on cognition and behavior. Most of them just put males and females together, but for prostate treatment and for breast cancer, you do get female only or male only. So for females in breast cancer, it does affect their verbal processing and so forth. And there's different types of chemotherapy. If it affects their verbal processing and facial processing and so forth, we'd expect that, or at least I would expect that. But if it also starts to affect their spatial abilities and other traits that aren't elaborated in them, then you probably have a very, very toxic treatment, and you may want to reconsider that. The approach can, as I said, can be used to develop associated assessments. And finally, we can bring the non-humans back in, and this might be especially true in a developing area like many parts of China. We have a lot of industrial pollution and other degradations of the environment. Deficits in sexually selected trait expression in other species, like some of the birds I showed you, for example, or butterflies or other insects, can serve as sentinels of environmental degradation. So if the males of, peacock male, we wouldn't get peacocks here, but if males that are otherwise very colorful, red, yellow, green, or whatever, if their plumage starts to become degraded, it's not developing as fully as it used to, you almost certainly have a severe environmental degradation of that area, which means that humans living in that same ecology are probably at risk as well for sex-specific deficits. And that's it. Sorry I went over a little bit.
May I now call upon Professor Lei Chang, moderator of today's lecture, to host the question and answer section for us. Professor Chang, please.
Oh, sure. Yeah.
So one question is, are there individual differences in terms of their vulnerability to this, sex-selected trait?
Right.
Like, for example, for the animal research, which is easier to kind of show, are the better endowed, males will suffer more heavy toll than the less endowed, or maybe it's the reverse, or maybe there is no difference.
Yeah, it's probably the reverse. So in naturally occurring populations, you have kind of a colorful plumage, so you get a bright yellow like the goldfinch there. All males will develop this coloration to some degree, but it'll be naturally distributed. You have malnutrition or parasite infestation. Some especially healthy males will only be affected a little bit on that trait, and other males will be affected a lot. For the whole group, you can have a shift in the mean coloration, but yes, there will be individual differences in sensitivity to these traits. So some males won't be affected that much, and others will be affected a lot.
Then does this kind of theory extend to just other health issues rather than this very extreme environmental stress? Like, for example, your overall health, our psychological health, our aging. When you experience that kind of more general, normal kind of, you can say, decline, do the sex-selected traits go first also? Yeah,
that's a good question. With normal aging, not Alzheimer's, but normal aging, the typical sex differences, say in verbal abilities that favor women or spatial abilities that favor men, those sex differences are maintained. It's only when you have something wrong that you begin to get these reversals.
Questions?
David, I'm very intrigued with your evolutionary research findings, because in promoting gender equality, we often say that we want to avoid sex typing, and we want to promote more androgyny.
Sure.
for both males and females. But no, with your result, it shows that you're exposing them to greater vulnerabilities.
Right. So I completely agree with gender equality in terms of opportunity, educational opportunities, and a variety of other things in the modern world. But that attitude of trying to promote equality is one of the reasons that none of these studies look at sex differences differently for, say, chemotherapy or other types of things. They put men and women together. So equality is a good thing, equality of opportunity is a good thing, but it's also led us to miss a lot of sex-specific vulnerabilities that we can actually do something about, but we haven't looked at for a variety of reasons.
Yeah, well, I agree with the gender-sensitive approach when we look at sex disaggregated data and research.
Right.
However, it's the conclusion whether we should be promoting more diversity or whether we should be promoting more sex-type type of behavior.
Well, I don't think we have to promote sex-type behavior, but because I think you're going to get that regardless of what you do. So if you look at boys' sex type, if you look at children's sex-type play, there's not much. Parents can squelch it a little bit, but as soon as parents leave, it goes right back to what it was previously. So you don't have to promote sex-type behavior. What I'm saying is, if you really want to understand vulnerable kids or vulnerable adults, you have to accept that there really are biologically based sex differences, and that we know what these are, we can assess these, and we can figure out where problems are in boys and men and where problems are in girls and women. But that's completely independent of educational opportunities and equality in workplace, and those are different cultural legal issues.
I am not a psychology student, but I think you at the beginning of your lecture, you said if a certain trait wouldn't respond to BPA, if you kind of assume it wouldn't respond to any chemical. How can you assume that it wouldn't respond to all chemicals?
Right. So if I understand correctly, so I'm saying that prenatal exposure to certain types of toxins or chemicals can have sex-specific effects on different traits. The way these studies are done in lab animals, we gave them pre-measured food, and so we knew exactly what they were getting. But in humans, the way they look at this is they look at maternal urine or amniotic fluid. They'll measure that at certain points, and you can actually detect this in breast milk. So if the kids are breastfeeding, you can pick up the toxins with that. And then what they do in those studies is they look at level of toxin levels in mom for however many toxins they're looking at, and they look at the correlation between levels in mom and the outcomes in the kids. And most of these studies have thrown boys and girls together, unfortunately, but some of them have separated out boys and girls and have suggested, as I said with the Taiwan study, that it's having different effects on boys than it is on girls.
Any questions? Raise your hand. I can't locate you.
So, David, well, of course, the boys and girls has a major difference in the genome, right?
Right.
the XY chromosomes. So from all the studies that you have mentioned, there seems to be not really correlating the genetic background, either the primary genome or the secondary genome, the microbiome, to the kind of studies. And we are looking at, or we are talking about at least as in the near future, we'll be looking at personalized medicine.
Yeah.
So perhaps sex difference may not be very important.
I think so. I think this has a lot of implications for treatments, medical treatments, so forth, for men and women, and maybe differentiating those. I didn't talk about epigenetics, and I think kind of how environmental exposure will influence patterns of gene expression. But I'm sure these traits are very heavily epigenetically dependent. But in my book, I'm not going to go to that level of analysis. So I haven't really looked at that yet. But I think that would be the next step. Exactly right.
Any more questions? If not, should we conclude here? And thank you, David, again.
Thank you, Professor Geary and Professor Chan again. To express the college's appreciation, we have prepared souvenirs for both the speaker and the moderator. May I now call upon Professor Jimmy Yu, college head, and Professor Helen Mann, chairperson of the Distinguished Visiting Scholar Committee, to present the souvenirs. Professor Yu and Professor Mann, please. First, to our distinguished visiting scholar, Professor Geary. And then to Professor Chang, moderator of today's lecture.